Bio-Chemist chapter here on campus. It stands for the Society for the Advancement of Chicanos and Native Americans in Sciences, and we work to promote sciences among younger students as well as create a network of science majors here on campus. Our most recent activity is starting a speaker series where we invited working professionals to come and talk about their education, their work, and their experiences. The first speaker in our series is Dr. Lino Gonzalez, who is a protein biologist at Genentech Labs. We are very grateful to have him here today, and without further ado, I'd like to introduce Dr. Lino Gonzalez.
So, there's a combination of the rain, there was an accident on Ottawa, and then hiking up the hill to get here. It was quite a trek. Anyway, I'm glad to be here. Thank you for inviting me. I was instructed to sort of give you a little bit of background on the science that we do, and also background on my career trajectory, how I got to where I am as a scientist, and maybe sort of outline some challenges along the way. And I want to also try to leave some time at the end of the session to answer questions, because oftentimes there's things that you have on your mind that maybe I didn't cover that I'd be happy to try to answer. So I'm going to start out with the science part. And this picture serves to illustrate to me sort of a lot of the fundamentals around why I got interested in doing science. Anybody familiar with this picture? Have they seen this picture before? Some of you? So this is Watson and Crick, and they're standing in front of a model of DNA. And it was in 1953 that they figured out, based on a number of scientific experiments and also intuition, what the structure of this DNA molecule looked like. And once they saw this structure, it clicked. It made a lot of sense. It explained a lot of biology. In particular, it explained how DNA from one cell could be replicated and divided and then form a double-stranded helix in each daughter cell. And that forms the basis of heredity, which had been understood for about a century, but not really understood at the molecular level how that happened. And when they had seen this structure, imagine that you're the only person in the planet in the history of humanity who understands this, who knows this. I mean, that is really powerful. That's something, it's an achievement which you can't really put a measure on. And so you could put yourself in these guys' place. They said when they saw the answer, we had to pinch ourselves. Like, they just didn't believe that it could be so elegant, so beautiful. So Watson told the BBC in an anniversary interview, could it really be this pretty? When we went to lunch at the Eagle, which is a pub right outside the lab where they were working, you realized that it was probably true because it was just so pretty. It just made so much sense. And so that encapsulates for me a lot of why I got interested in science. Right? Because science is really discovery, it's really understanding things that we don't understand, especially in biology about ourselves and how life functions. That was really my inspiration. And many, many years later, this is about 50 years later now, this is Watson from the same picture. He visited our company, he gave a seminar, and I took a couple of my books, a first edition of his double helix book, and had him sign it. So it's on my bookshelf, and it's a great connection between, sort of this history and having someone who visited my company and signed this book for me. So this figure serves to illustrate the link between DNA, chemistry, and life. And this was some of the fundamentals that really inspired me. I started as a chemist, as an undergrad. I'll get into that when I get to the career section. But it was when I realized that chemistry sort of underlies, in very basic ways, life, and consequently us and how we function. So this illustrates the connection between the two. And the other thing that sort of got me more into the biology field from the chemistry was really being fascinated by cell biology, molecular biology. And this is a beautiful picture of a couple of cancer cells. And here you can see that they're They have some kind of communication going. There's a lot of invisible things that are going here. These cells are secreting proteins from the cell surfaces. They can diffuse through and they can bind to receptors on the other cell, and those cells can recognize those factors, and they can lead to cell growth, cell proliferation, cell migration, a lot of physiological events. But this process is incredibly complicated. It's so complicated, we really don't understand it. We're just starting to understand it. When Watson and Crick identified the structure of DNA, and then a few years later had sort of decoded what's called the amino acid sequence that translates the DNA to a protein, they thought that it was done. Basically, they figured out everything there was to know about cell biology and how cells work. And that was further from the truth. And I'll get into a little bit about how that is. But this picture serves to illustrate that. So here's a person looking at a problem, maybe looking at a small portion here, but in fact, the problem is huge, it's immense. It's bigger than any one person can really even understand. Okay, so it's interesting how you have a cell which is microscopic, right? You have to look under a microscope to see it. But there's so much complexity there and so much going on that you can't comprehend. You know, just as a person, you can't understand everything there is to understand. So really, as a community of scientists who are studying life science, it's all of us working together to try to study different aspects of cell biology and make sense of it, integrate it so that we have an understanding of how cells work. And of course, cells make up us and all other organisms. So to sort of dive down into, just to get a sense of this complexity, I'm showing a representation of one of the chromosomes in humans. So obviously, our DNA lives in these chromosomes. And on these chromosomes, there's a number of genes. Okay, I'm sure you've all heard of genes. There's hundreds of them on this particular base, on this particular chromosome, and this is made up of 135 million base pairs. So you're probably all familiar with the DNA base pair structure, right? AT, GC, DNA. Well, each base pair is one out of 135 million that make us this one chromosome. Okay? And all this information, plus all the other chromosomes, reside in every single cell. So every single cell, with a few exceptions, in your body has the full complement of DNA. There's actually more than 3 billion base pairs in each cell. So if you think about it, that's a huge amount of information. People think of it, well, okay, if you equate the base pairs in a DNA to sort of a letter in a book, right? I don't know how many books. It would take volumes and volumes of information of books to fit all that information into. And all of that is in one of those little cells. But becoming even more evident, and this is very, very, very recent stuff, that's just emerging in the last year or so, is an additional complexity of how these genes are regulated. So not only are there genes on these chromosomes, there's regions in between the genes that are very essential in regulating how these genes are made and when they're made and why they're made. So in response to something the cell perceives, it can change its genetic program to express certain genes and turn down certain genes. And this is a whole dynamic between genes that are expressed, genes that are not expressed, and the functions of those genes. Okay, and you got to think of it as a living system. So these things are going up and down all the time. And it's really a program that's mediated through these intervening sequences between the genes that allow them to be regulated up and down. This all gets into the connection with stem cells. You've probably heard of stem cells, and how stem cells are sort of very basic cells of a tree of diversity. These cells can then differentiate, which means they can turn into different types of cells depending on the genetic program that's initiated. Okay. So obviously I'm going to leave it at that, but you can get a sense of just the immense complexity that's involved. This is a picture of the Wellcome Trust Sanger Institute at the UK. So I had the honor of being able to give a seminar there last December, and it's a fabulous place. The one thing I want to tell you about is that this was one of the institutes where the human genome was sequenced. The other was the National Institute of Health here in the U.S. But they used, a lot of high throughput instruments to sequence, all 3 billion base pairs of the DNA sequence. Okay, and this was done about 12 years ago now, and it was a huge breakthrough, right, for scientists studying life science, because then you have the book there, and then you have to make sense of it. Again, to give you sort of a sense of the magnitude of the information, when I was walking through the hallway there, they had this poster. And if you look really close on that poster, there's these little APDCs, you can see? And that makes it the whole poster. And it's actually a printout of a part of the X chromosome. So someone on the computer actually decided to try to print all this out. And so there's a little page right here, and if you read it, it says, this banner contains 1,346,400 bases of DNA sequence from the human chromosome X. The entire X chromosome sequence was printed on 115 of these banners for a certain scientific meeting. So if you take 115 of these and align them on the wall, that's equivalent to just the X chromosome. Okay, all, the rest of it, that's just a small portion. So it really is an incredible amount of information. Okay, so another huge thing that's happening in science right now is the ability to sequence DNA genomes is advancing so quickly over the last 5-6 years. It's gone through the roof. And so where it took billions of dollars to sequence the first human genome, it's really becoming, within the foreseeable future, going to be possible to sequence a full 3 billion base pair human genome for $1,000. Okay, and that just changes what you can do, the questions you can ask about in science. This is sort of equivalent to, I don't know if anybody's familiar with Moore's law of computation, where it says, every few years the price of computing, decreases and you increase the capacity and the speed of computing. Similar thing has been happening in sequencing. It not only has consequences for human health and our understanding of disease, but it has huge implications for our understanding of the relationship of life on the planet and also how life has evolved and the interrelationships of different species, because not only can you sequence human DNA, you can sequence any type of DNA from any organism. So it's been huge to those fields. And so you can see here the number of human genomes that have been sequenced now is approaching the tens of thousands, and this is going to continue to increase very rapidly. This is really enabling some unique types of studies where you can look at populations of people. And one of the coolest things I've seen very recently is that if you take the genomes of those people and you look at their disease, what kind of disease they have, say Alzheimer's disease, there's some people that if you plot them out on a bell curve that get very early onset Alzheimer's, and then some people that get very late set Alzheimer's. So obviously there's some kind of genetic component that predisposes them to having it a very early or very late. And so what bioinformatics specialists can look for is they can analyze all these DNA sequences, and they can compare the DNA sequences from those that have